all the way from the University of Sydney. And from what I know of Peri, she is an amazing person. And she does a lot of genetics, which I do not understand most of the time. But we all really appreciate my input to this. So over to you.
Thank you, and thanks for the organising committee, particularly Jenny, for organising everything here. It's been sort of blown my mind the how this Australasian Myrtle Rust conference has kind of involved. It's wonderful to see, and the research and the presentation have been incredible today. So I'm talking about the insights into the genome biology of the Myrtle Rust fungus, Australasian and genome biology and evolution. And that is a plant just down the road from where I live. I live in Manlyvale, near Manly, and that on my regular walks, I just take lots of photos of infected plants. That's what you see in my region. So you see a lot of infection. And I wanted to, almost a segue from that voting for the fungus, just want to remind you that it is a pretty amazing organism, like it's doing some terrible damage, but the biology of this little microfungus is incredible. And so that was just an image that I took through the microscope lens of a germinating spore, and it's very fetus-like. That's kind of a nice picture. But just to remind you, we're talking about a devastating pathogen for the plant, but it actually, has a life of its own, which is also quite interesting. Genome resources just keep improving, and that's because of sequencing technology and the chemistry and the software is all just very, very rapidly changing. So we've got 3 iterations of the actual genome for this pandemic strain of the fungus. The first, which we published in 2021 with a lot of our collaborators in New Zealand, what we found that this fungus was a very, very large genome, which is quite unexpected. And in fact, it's 3 times the size of the eucalyptus host genome. So it's pretty amazing. The eucalyptus grandis grows to 50 metres, and this little fungus is, you've seen it, it's tiny little spore. It's the size of it's due to this massive transposable element aspect of the genome. So these are kind of regions of repetitive DNA which jump around the genome. So that was a good finding. And the next iteration of the genome, based on some new data, we were able to actually scaffold into chromosomes. We found that it actually had 18 chromosome pairs, and we were able to assemble a complete mitochondrial sequence and also identify the mating loci, so how the actual pathogen mates, with other pathogens, with other reservoirs. And now we've improved it even better. And so this latest publication, which is still a preprint, is largely the work of Jenyan Liu, who's been a PhD student under Benjamin Schweiziger at ANU. And we've had some more very interesting biological findings from this. So I'm going to talk a bit about her PhD work. So some of it I don't understand, but I'll try. But before I do that, I just wanted to also say why is this important that we get a good genome, and sometimes it seems that, we're doing science and we really are just interested in the problem we're trying to solve in science, and maybe it's hard to see an application. So many times we are actually not thinking of an application. We're really just trying to understand this organism. But what sometimes arises is a practical application just from our sort of blue sky type research. So in 2022, once we've been able to identify the 18 chromosomes, we could find these mating genes on her chromosomes. And that was quite a good breakthrough because it allowed us to, if you look at there are kind of 2 types of mating genes. And if you look on the in the red boxes, in this first column, you'll see a ACDI from Australia, South Africa, Brazil. And you can see that the little tree that they're sitting on shows that they're actually divergent. You know, they're a little bit different. Whereas the genes, the other mating genes from South Africa, Brazil, and the pandemic show that they're actually exactly the same in the other boxes. And so that kind of gave us a clue that our strain that we have in New Zealand and Australia is a little bit different, you know, to the other ones. And we want to keep the other ones out. So maybe we can use this to identify as a biosecurity thing. And so that's what Ash was talking about just before. We worked on developing a diagnostic to identify this using long-read sequencing, and it's quite successful. So we've been able to show that. And then from that, the Australian government have now commissioned us to write a new diagnostic protocol to identify strains, so which Ash is involved with as well. So this is all in progress right now, and if it's helpful to us, maybe it'll be helpful to you guys in New Zealand as well. So we didn't go out with this objective, right? We had no idea that we would find this, but we found it. So sometimes you just leap in and try and identify things and something arises. So that's me justifying why we did genome work. So we have this new revised genome, and what makes it so good is we were able to incorporate long read data from Oxford Nanopore reads, along with our other hi-fi data that we already had, and we used our Hi-C data. So these are 3 different types of sequence data that really together brought this to, you know, really improve this genome. And not only that, but Jan ran all the hi-fi data through Google's Deep Consensus software, which actually improves the accuracy of those reads before you actually try to even assemble them. And then we annotated, so we looked for all the proteins. We annotated those using both short read and long read RNA data. So this is far more, you know, gives us a far more better picture of the actual genes in the pathogen. And then we did some cytological studies. So the studies were, you know, that's actually been quite a difficult thing to resolve in Rust fungi, to do this sort of visualizing chromosomes that you can see in the top image. So we were able to see from that 12 of the chromosomes are quite large, 6 are quite small. So that we can actually see this first time we've been able to see these chromosomes. So that was very exciting. And then, as I say, we identified that they had 18 chromosomes and they're conserved with wheat rusts. So even though they're very distantly related, they share something. These graphs, the graph on the right is a heat map showing how we build the scaffolds, the chromosomes using the data. And if you look at the little blue boxes, you can see that sort of bow tie sort of shape. Those are the chromosomes. So the data just allows us to identify the chromosomes. So what we did was and you can see that there are 2 boxes, and that represents the 2 nuclei. So Myrtle rust has 2 separate nuclei inside the spore, and we can actually see that with the data. It's very, very clear. But what we did find was that chromosome 14 appears twice in one of the nuclei and doesn't appear at all in the other nucleus. So that was really surprising. And we did this many, many times, and we're pretty sure this is correct because statistically it just every time falls out like this. We also did some protein homology work. So looked at the protein on the left. These are the chromosomes. So from haplotype A and B, the top one is chromosome 1, the next one is chromosome 2, all the way down. Chromosome 14 looks a bit different when we look at the proteins, right? You can see sort of dense pink lines there. That means they're more closely related. A sort of explanation for this might be that a long time ago, perhaps back in South America, this original rearrangement of the chromosomes happened. And DNA has a way of repairing itself when there's damage in, it uses the matching chromosome pair to try and fix itself. But because these chromosomes are separated in separate nuclei, they can't do that. So at the moment, what's happening on an evolutionary scale is they're becoming more and more different. But the chromosome 14, they're becoming more and more similar. So perhaps, you know, you could speculate that this has some meaning, you know, for the spread of this pathogen. We can't really say that for sure, but it's just interesting. So as I said, we've just published this preprint, and there are a number of papers there, and I'd just like to attribute a lot of the research was done by Jen Liu. And those are some of her images from her PhD, and just some germinated spores on the leaf surface and some timed fluorescence of some of these spore developments. So 3 days after inoculation, you can see the hostorial structures developing. At 6 days, the spore, the pustules developing just at the surface of the axial surface of the leaf, and then at 9 days, you've got the actual eruption of pustules. So all those are going to spread further. So with our new improved genome, we thought we could ask a few more questions, and we asked, well, does DNA methylation play a role in the transposable element silencing? Because these jumping regions of DNA are usually a concern for most organisms, so don't like being mutated too much. And then what is the genome biology of the mating type loci? And does the pathogen have a distinct infection progression in different susceptible hosts? And we also thought we could look at the allele-specific expression. Turn my timer on, but
You got 5 minutes.
okay. So this part of the work, which shows the how much of the genome is covered in transposable elements, is interesting. So on the right, you can see those are different classes of transposable elements, and they just about cover each full chromosome. So the rest of the regions are actually the genes, and there's not much of that. So it's mostly transposable elements. We didn't find any of these classic repeat-induced point mutation signals, but we did find DNA methylation genes were there. And we also found that over 90% of the methylated CPG sites overlap with transposons. So this is likely to be a method to silence the transposable elements. And then over time, those methylated cytosines can transition to thymines, which is what we see in the actual genome ratios. As far as the mating genes go, we could see very clearly with if you look at B and C graphs, the blue bar is where the chromosome, where the centromeres are on the chromosome, and just to the left of that is a ST3.2 and an ST3.23, and those are the genes, the receptor genes for mating. And it's hard for me to point to this, but the regions just in front of those genes for mating, the receptor genes, there are no other genes. So the red little lines, the red lines on the gene density show you how many.
Am I in the right place?
These are the red lines. This shows gene density, and you can see there's a big gap just next to the line marking where the pheromone receptors are. And this is classic for recombination suppression. So this is when you see sex genes, we don't want them to mix. We want them to stay very, very distant, be different, so that you get them, you know, for example, male, female, plus minus, the genes have to remain quite different. So that was good to see that signal there really is very clear. Again, we also saw a very unusual expansion of these pheromone receptor peptides. So, and we haven't really explained that because it's quite, we don't see that in other rusts. So it needs further work. As far as the temporal infection, using we got 4 susceptible species: Syzygium jambos, Leptospermum scopareum, this is actually an Australian one, Melaleuca quinquenervia and Melaleuca alternifolia. They all develop pustules at 7 days post-inoculation under controlled infection. So if these were screened prior to this, we knew they were susceptible. So we chose susceptible plants, and the timing was exact. So that was interesting. And we also looked at some gene expression. And if you look at the PCA plot up the top, you can see the black dots in the left corner at 0 days post-inoculation, before inoculation. The other dots at the top are the early infection stage. And then on the right, that's the late infection stage. So we've got very distinct patterns of infection genes that are being expressed. And I guess the next thing just to point out is the shared candidate effectors. So these are I'm talking about the pathogen's expression, right? Not the plant. So the effectors, which are the secreted molecules that initiate infection and control the host. When we look at the haplotype-specific genes that are expressed at 2 days, 3 days, 4 days, 5 days, and 6 days, the 2 lines you see at each of those, it's hard for me to point, I guess what you can see is that there's a different pattern in column 1 to column 2. That's haplotype A and haplotype B. So what we're seeing is each of those nuclei is expressing different genes at different times. So that's very interesting because it means, you know, they have different genes. They've evolved slightly differently. So they're in 2 different nuclei. So we can see that very, you know, that's really just what that graph is showing. There's a lot more to this, and in the paper, if you want to read the paper, it's probably better. But the key overall findings, so we do find distinct methylation patterns, profiles, and the recombination suppression of the pheromone receptor genes, and also the expanded peptide pheromone precursor genes. We also see conserved infection dynamics in the susceptible hosts and 2 distinct waves of gene expression in early and late infection and the allele-specific candidate effector expression. Big spores there. Just some acknowledgements, and I really hope I haven't missed anyone off, but this is really just the authors of the latest publication that I'm acknowledging here. And any questions that you have, you can email me here. And I also just wanted to mention that I just put this other genome up to be public, and just because it might be useful, it's a Syzygium lemannii, so that's a phased genome for that plant, and it's just a preprint. It's really a genome report, having done further analysis, but what's interesting is it's a susceptible Syzygium. So now we have Syzygium myri, Syzygium lemannii, and it seems we've got Syzygium jambos, which is tetraploid. You know, the actual susceptibility of these plants is also something that needs to be investigated, not just the resistance in resistant plants. We need to know why these plants are susceptible. And there's obviously targets that the pathogen is finding in these plants. So that's now currently available, all that data. We plan to do more work on this. It's just at the moment, I just thought I want to make it public. So that's it for me.